It was a birth announcement that shocked the world.
A researcher in China says he worked in secret.
An experiment aimed at protecting children from ever contracting HIV.
The reaction was shock and condemnation.
Because we just do not know the risks, and the risks could be severe.
In 2018, a man made a shocking announcement. What followed became one of the biggest controversies in modern medicine. Some hailed this man as a hero who could save thousands. Others warned that he could change the genetic code of humanity forever. And let me tell you, I had no idea how deep this rabbit hole goes. This is the story of the first genetically edited children. This is the gene editing scandal that shook the world. An underdog is, quote, the weaker of two competitors or anyone not expected to win a competition. The story of the underdog overcoming obstacles against all odds is a story often depicted in cinema. We often find ourselves cheering for the underdog, celebrating their victories. But what if the determination of the underdog gets distorted and they act unethical or possibly dangerous? Well, this might be the story of He Jiankui. He Jiankui was born in 1984 in a small village in Xinhua County, Hunan, China, into humble circumstances. His family lived in poverty and the whole village lacked electricity. Despite these challenges, his family recognized his exceptional intelligence and did everything they could to support his education. He was determined to excel academically, and he studied day and night. His hard work paid off when his perfect grades secured him a place at Xinhua School in a program for gifted children. After finishing high school, he enrolled in a physics program at the University of Science and Technology of China and conducted his PhD research at Rice University, Houston, US. During his PhD research, he used statistical models to study different systems, including global financial markets, emerging strains of the influenza virus, or animal anatomy. However, after his PhD, he felt that his ambitions were better suited for another research field. A field that could change the world forever. In 1996, scientists at the Roslin Institute in Scotland achieved a revolutionary breakthrough. They successfully cloned an animal. A tiny baby sheep named Dolly opened her eyes. Dolly was a clone that had been created for a technique called somatic cell nuclear transfer. Somatic cell nuclear transfer means that we extract an unfertilized egg cell from a sheep and removing its genetic information, or DNA. Then DNA is taken from a mammary gland cell of another sheep and transferred to the egg cell. Through a series of carefully controlled steps, the egg cell is then stimulated to develop into an embryo that is then transferred into a mother sheep. In this way, a sheep is born that contains the same DNA as the sheep that donated its mammary gland cells. In other words, a genetic clone is born. Dolly was a huge success and a milestone for regenerative medicine. Media outlets all over the world reported about the cloned animal, and countries started to pour in billions into regenerative medicine in a race to achieve the next milestone. One of these countries was China. Between 1992 and 2019, China's investment in research and development has increased from $2.2 billion to $305 billion. Research programs in China were developed to recruit talents who are conducting research in other countries. And so in 2012, He was recruited by the Southern University of Science and Technology in Shenzhen, China, to open his own laboratory. Once more, He worked as hard as he could. He successfully founded multiple startups and was celebrated as a pioneer of genome technology. But then he did something odd. Using the money he earned from his companies, He took an unpaid leave from the Southern University of Science and Technology. But he wouldn't take vacation. Instead, he was preparing something big and secretive. April 2017. A man sits down to read a newspaper when an advertisement catches his eye. A free experimental fertility treatment for HIV-positive patients. The man was shocked. You see, he was HIV positive, but he was taking his medication and his wife and him wanted to have a child. They decided to give it a shot and responded to the advertisement. He filled out a questionnaire asking basic questions about his age or if he's married. But then there was also a very specific question. Would you be interested in learning more about a gene therapy that could reduce a children's risk for becoming infected with HIV? A couple of days later, he received a phone call. It was an assistant of Xinhua He. The assistant revealed details of the fertility treatment. He and his team aim to genetically modify embryos to become resistant to HIV infections. He has gained a reputation to be at the forefront of a novel gene technology called CRISPR Cas9. CRISPR Cas9 is a technology that allows precise changes in the genetic information of a cell. To do this, we need to do the following. We need to introduce the Cas9 protein and a small piece of RNA into our target cell. Cas9 is a protein that cuts DNA, and the RNA has been designed to guide the Cas9 protein to the right location. Once introduced, the Cas9 protein and the RNA form a complex that starts to scan the DNA of the cell. When the Cas9 RNA complex encounters the right DNA region, the Cas9 protein makes a cut. The cell senses the cut, tries to seal it, and in this way causes changes to the DNA sequence. Sounds complicated, but the important take-home message is that if we add an RNA with the sequence of a gene X and a Cas9 protein, then the Cas9 protein will make a cut in gene X. Using CRISPR Cas9, we can try to change any gene we like. He and his team aim to target the CCR5 gene and cut out a 32 base long sequence. In this way, they want to introduce the so-called CCR5 delta 32 mutant in embryos. But why should they do that? Well, the CCR5 delta 32 mutation is naturally found in a small percentage of humans, and it makes them resistant against some HIV strains. By introducing the CCR5 delta 32 mutation in embryos, HIV particles become unable to enter the cell and the resulting baby becomes HIV resistant. That's their theory, at least. The man was amazed. He immediately wanted to enroll into the clinical trial. There were only 3 conditions he needed to meet. Number 1, his HIV viral load must be under control, meaning that he needs to take his HIV medication. Check. Number 2, he needs to have fertility problems. Check. Number 3, he needs to have a certain educational level and maintain open communications with his doctor. Check. The research team then explained all possible risks. This procedure is unprecedented and might have severe side effects for the unborn child. Still, the man and his wife agreed to the procedure. He and his team also enrolled 6 other couples for the trial. All participants signed an agreement pledging strict confidentiality. They were not allowed to talk to anyone about this trial. This was a bit odd, but there was a good reason. Fertility treatments for people with HIV were forbidden in China. So the trial needed to be treated as a secret, at least for the time being. The procedure began. He's team isolated egg cells and sperm cells from the couple. The egg cells were then fertilized in a petri dish. Moments after conception, He injected the eggs with Cas9 and an RNA against CCR5. The embryos were then put into a warm incubator and some cells were taken for testing. The DNA sequences of the embryos were then analyzed. Overall, 31 embryos were genetically modified and the CRISPR Cas9 editing worked in 70% of cases. These were good results, but some of the embryos showed alarming genetic changes. Keep that in mind, we will come back to that later. The most promising embryos were transplanted back into the mothers. Now they only needed to wait. October 2018. He and his team rushed to the hospital. Soon, their first genetically modified babies, a pair of twins named Lulu and Nana, would be born. The birth was complicated. The twins were born prematurely by emergency C-section, but they seemed to recover quickly. The parents, He and his team, kept the gene therapy a secret from the hospital in fear of national news. They wanted to wait until the health of the twins stabilized. November 25, 2018. Somerville, Massachusetts, US. Antonio Regalado sits at his desk. He's just finishing a story on CRISPR Cas9 research in China. Over the past weeks, he interviewed several researchers, including Xinhua County. Regalado googled his name once more and found a new document. A clinical trial on CRISPR Cas9 gene therapy to prevent HIV infections in embryos. The document has been uploaded to the internet a couple of weeks earlier. Regalado was shocked. He didn't know about this. So Regalado called He, who was on his way to an important conference. The scientist brushed him off. Regalado quickly wrote an article and posted it to the MIT Technology Review website. Exclusive: Chinese scientists are creating CRISPR babies. A couple of hours later, another study was published by a team of journalists who had been investigating He for months. He saw the articles and realized that he had to make his research public. November 26, 2018. He told the organizer of the second International Summit on Human Genome Editing in Hong Kong that he had altered the DNA of 2 embryos. He wanted to present his findings there in 2 days. November 28, 2018. He enters the stage. Among the audience were some of the most esteemed researchers in the world. With a dramatic flair, he announced the birth of 2 genetically modified babies. He felt, quote, proud and noted that a 3rd gene-edited baby could soon be born. His goal? Not to cure a genetic disorder or prevent one. Instead, he wanted to make babies that are resistant against HIV infection. He presents some of his preliminary results in some experiments, but keeps details scarce. The audience was silent, then irritated. Some researchers asked questions, others criticized He's lack of transparency. Others remarked that he didn't make any safety precautions. He answered some questions and said that he will officially release his results soon. Overall, his announcement was polarized. Some celebrated him, others were shocked. Developer of the CRISPR Cas9 technology and Nobel Prize laureate Jennifer Doudna was quoted saying, "I was just horrified. I felt kind of physically sick." The news spread like wildfire. Chinese social media was flooded with discussions about the genetically engineered babies. Some were concerned about the baby's health, others said that this was a violation of the laws of nature. As a form of damage control, Ha's team released the signed forms of consent and a YouTube video. There he explained in greater detail his visions and goals. But that didn't really help. He was tracked down by reporters again and again and again, but soon he would vanish. But why were scientists so shocked by He's announcement? Well, you see, in the past, first attempts at gene therapy had resulted in some catastrophic outcomes. In 1999, a patient with a rare genetic condition underwent gene therapy, but tragically passed away shortly afterward due to severe immune responses. In another clinical trial, children with XKID who underwent gene therapy were successfully cured of their disease, but 2 of them later developed cancer. In these cases, genetic modifications were involuntarily made in other DNA regions that then caused the cells to slowly transform into cancer cells. And this is why gene therapy is heavily regulated. While the concept of gene therapy may seem straightforward, we just put in genes and remove genes, there are numerous factors that can lead to harm for the patient. And since He has not released his findings yet, no one knows what safety precautions he took. But there were even more issues. Normally, gene therapy is only performed in adults suffering from a severe genetic disease. In this case, only a small number of cells are genetically modified, and if something goes wrong, the genetic changes remain confined to these cells. However, He modified embryos, meaning that any cell could carry genetic changes. These include egg or sperm cells, and that is bad. If egg or sperm cells are genetically modified, the genetic changes are introduced into the gene pool. This is concerning because, once again, if the clinical trial were to fail, future generations could be adversely affected. November 29, 2018. Reports surfaced that He had forged documents, including the approval form of a medical ethics committee. The university where He conducted research released a statement saying He's experiments was done, quote, outside of the campus and was not reported to the university nor the department. He sent his manuscript to 2 publishers, but it was immediately rejected. Some speculate that this was due to the suspicion of forged ethical approvals. The Chinese government stated that it had suspended He's work, adding that his behavior seemed to violate Chinese law. Another presentation of He that had been scheduled on the same day was cancelled. He then seemed to have been put under some form of surveillance. Rumors surfaced that He had been apprehended by authorities, which was denied, but the scientist wouldn't be seen for a long time. January 21st, 2019. For forging documents, He was officially arrested and he was formally and publicly charged. A day later, President Xi Jinping called for new gene editing legislation. Additionally, a statement is released that the health status of the born twins is being monitored. July 2019. A 3rd genetically modified baby had been born. There were no further details released. It is not clear what happened to the other 5 couples that had been part of the clinical trial. December 3rd, 2019. The manuscript that He attempted to publish back in November 2018 was leaked to the internet. The following section is based on an article by Antonio Regalado, who interviewed several experts in the field. Finally, the complete truth came to light. Disclaimer: although this document is most likely authentic, we cannot verify the accuracy of her statements. In the first paragraph, the document states that, quote, we use CRISPR Cas9 to reproduce a prevalent genetic variant of the CCR5 gene in fertilized oocytes during in vitro fertilization procedure, with the aim of helping the twins to be born with their own natural protection against HIV. Genome sequencing during pre-implantation genetic testing and after birth confirmed that the twins' CCR5 genes were edited successfully and are thus expected to confer either complete or partial HIV resistance. No off-targets, larger lesions, or pathogenic cancer mutations was observed. Here we bring a novel therapy to enable acquired immunity to HIV and control the HIV epidemic. Sounds very sciencey, but there are 3 key conclusions from these statements. A. He and his team successfully introduced the CCR5 delta 32 mutation in both twins. B. There were no unwanted genetic changes elsewhere in the DNA. C. The treatment holds promise for controlling the HIV epidemic. Let's examine each conclusion individually to assess whether He is correct. Humans have 2 copies of most genes, called alleles, with one inherited from the mother and one from the father. In order to be considered HIV resilient, every cell in the body needs to have the delta 32 mutation in both CCR5 copies. However, later in the document it says, quote, one embryo was edited on both CCR5 alleles, with each containing frameshift mutations that deactivated the CCR5 protein. We expect this to confer complete resistance to HIV-1 virus infection, similar to the natural CCR5 delta 32 mutation. The other embryo had one allele edited with a 15 base pair deletion. One of the twins has mutations in both CCR5 copies that are quote, similar to the CCR5 delta 32 mutation. The word similar is concerning as it means that the gene editing didn't work as intended, resulting in a mutation different from the desired CCR5 delta 32 mutation. Moreover, the other twin had a different CCR5 mutation in one gene copy, while the other gene copy was not edited at all. But there is more. Sequencing analysis in a document shows that not all cells carry the genetic changes. As a result, some cells were genetically altered, while others were not. In other words, they are probably not HIV resistant. Now to the second statement. There were no unwanted genetic changes somewhere else in the DNA. He uses the word off-target to describe unwanted genetic changes somewhere else in the DNA. And that is actually very important. It is important to avoid off-target effects to prevent adverse effects we have observed in the early gene therapy trials mentioned earlier. In the beginning of the document, He claimed that they didn't detect any off-targets. However, later he states, quote, the other embryo showed one possible off-target of a base pair insertion in an intergenic region on chromosome 1. This means that the Cas9 treated most likely introduced an additional DNA base into another chromosome. He argues that this is not important as the change didn't occur in a gene, but it is also something dangerous, and a direct contradiction to the first page of the document. The third claim, the treatment can be used to control the HIV epidemic. The main argument here is that we can make large pools of people resistant against HIV. However, the CCR5 delta 32 mutation He aimed to create only helps against some HIV strains, not all of them. Moreover, we have several tools to combat HIV, including condoms, PrEP, or antiretroviral therapy. Of course, you can and you should criticize that not everyone has access to these treatment options, but they are much more likely to help than injecting millions of embryos with Cas9 proteins to make them resistant against some but not all HIV strains. And this is what most people on social media get wrong about He Jiankui. He was not successful in creating HIV-resistant embryos. He was not transparent about his work, repeatedly contradicted himself, and used gene therapy in a way that is not justifiable. Anyway, the story doesn't end here. December 30th, 2019. The court concluded that He and two of his colleagues violated regulations, practiced medicine without a license, and crossed an ethical line with the use of the Cas9 gene editing technology. He was sentenced to 3 years in prison. The two collaborators received a 2-year and 18 months prison sentence and a fine. The academic board of the university's biology department, where He conducted his research, said that He's experiments, quote, seriously violated academic ethics and codes of conduct. It was one of the biggest research scandals in modern China. However, shortly afterwards, the scandal faded out of the national consciousness. It seemed like censors were systematically removing posts about He and his clinical trials. As the country went into lockdown with the emergence of COVID-19, the scandal faded away. April 2024. He has been released from prison 1.5 years earlier. And he's back with news. He opened a new laboratory in China. Yeah. According to him, Lulu and Nana and the third genetically modified children are all still healthy. For some, He is a hero who bravely ventured into a medical frontier exploring a gene therapy that could potentially eradicate genetic disease and save millions of lives. For others, He is reckless and unethical, endangering lives merely to secure a place in the history books. Gene therapy might save numerous lives one day. So once it has been implemented, there are some questions. Where do we stop? Would we cure fatal genetic disorders? Most likely. What about less severe diseases like allergies? Maybe. And what about favorable traits like height, weight, or eye color? But what is your opinion? At this point, I'd like to give a shout out to the book The Mutant Project, Inside a Global Race to Genetically Modify Humans by Eben Kirksky. This is not a sponsorship. The book was incredibly helpful to me when I started researching for this video, and I think you might find it interesting too. With that, I'll see you. If you're interested in controversy involving creating stem cells in a petri dish, or a surgeon who endangered his patients to gain fame, you might like these videos.